Hey everyone, let's take a look at 16.5 for the lac genotypes shown in the accompanying table. Predict whether the structural genes are constitutive, permanently repressed, or inducible in the presence of lactose. And so this is the table that we're going to be working with, but I think we should probably review the lac operon first to make sure we're all caught up with how that works. Okay, get my pen here as usual. Okay, so let's just quickly sketch the lac operon. Now this is just a series of genes that is used to express the catabolism genes for breaking down lactose when lactose is present in the environment. So this is going to be LAC-R, so the repressor gene, and it's not part of the operon, so that's why I indicated that with those little hash marks here on our chromosome. Okay, we have the promoter here, then we have the operator. Let's call this P, and we can call this LAC-O. Sometimes it's called LAC-O, sometimes it's just called the operator. Then we will have LAC-Z, LAC-Y, and LAC-A. So these are the structural genes of the operon. Remember, a structural gene is any gene that does not encode a protein that regulates expression. This is the beta-galactosidase. This is the permease that lets lactose into the cell. And this is some kind of enzyme, and we don't quite know or understand why it's part of the operon. We don't know its role yet. And when I say we, I mean scientists. So, okay, so here's our lac operon, and how does it work? Well, the operator has a little binding site, so a little binding site here, and this is a DNA sequence, a specific DNA sequence that is recognized by the lac repressor. And so the lac repressor normally looks like this. Okay, and we have a little, this is our binding site in the protein. So the LAC-R protein, the repressor, can bind the operator when lactose is not present. So here's our lactose, and you can see it, kind of fits right in here. The way we've diagrammed it, it can fit right in here, and that's conceptually like how it works. So if lactose was present, it would fit right here into a little spot on the LAC-R protein, and it would cause an allosteric shift, which is a change in protein shape, and it would pretty much get rid of that site in that LAC-R protein, which has high affinity for the repressor binding site on LAC-O. So okay, so I think that's how the lac operon works. So lactose is okay. So when there's no lactose, this guy right here, repressor, will be bound over here. Okay, that makes sense. Okay, and then RNA polymerase can't come through here and can't transcribe the structural genes which we would use to break down lactose. Okay. Now there's a whole, we're not talking about the cap binding protein, which binds to the part of the promoter region up here, the cap binding site. We're not going to talk about that part in this question. We'll save that for a later question. And that's the cap protein. So let's not worry about that for now. Let's just worry about the repressor. Again, okay, so no lactose around. the LAC-R protein binds to the operator and polymerase cannot transcribe the structural genes. And okay, so lactose is present. So what happens? Okay, lactose will bind here, and then that gets rid of this binding site here, right? So I can erase all of this. Okay, I'm erasing this, erasing. Okay, and I can sketch this again. So there's that, there's the binding site. Okay, and there's our LAC-R. See, it can't bind. So lactose is present, can't bind. RNA polymerase comes in, makes the catabolism genes, and especially this one right here, beta-gal, takes lactose and breaks it down into galactose and glucose. Okay. Okay, so let's take a look at the question now. Okay, so we want to look at each of these conditions, and we want to determine, predict whether the structural genes, and here we're just kind of looking at Z, I guess it doesn't matter if we look at the others, if Z is expressed, the others are expressed too. Determine if the structural genes are constitutive, permanently repressed, or inducible in the presence of lactose. And we can see the first one here. The first one is, okay, if we have a wild type copy of I. Did I say, I've been calling this LAC-R, this is actually LAC-I. Sorry about that. Okay, if you call it LAC-R in the exam, I don't care. That's fine. Let's call it LAC-I instead to be consistent with the problem. Okay, so we have a wild type copy of LAC-I. Okay, wild type copy of the operator, LAC-O, wild type copy of the structural gene. Okay, this is a normal operon. So in the presence of lactose, the operon is going to be turned on, LAC-Z is going to be made. So this is an inducible genotype. In this genotype, LAC-Z, or the structural genes, are inducible. Okay, now how about this one right here? So in this one, the second one, we have LAC-I, which I was previously calling LAC-R. Sorry about that. LAC-I is mutated. Okay, so mutated, and now we're calling this specific mutation a mutation in the operator binding site. So I guess we just have to memorize that, because there's another mutation that we could have, which we'll talk about in a second. But the LAC-I mutation means we have a mutation here. Okay, so the operator binding site has been mutated. And because LAC-I can never bind to the LAC operator, well, RNA polymerase can always bind to the promoter and can always transcribe these genes. So this one is going to be sorry about that, that's a difficult word, constitutively expressed. So constitutively expressed, because it's always going to be on. That's what constitutive means, always on. It's always being expressed. The structural genes are always being expressed. Okay, let's take a look at the next one. Here we have sort of the same situation. We have LAC-I is mutated in the same way, cannot bind to the operator, and the LAC operator has a C mutation here, and C here. Now when we see LAC-OC, what this means, you kind of have to memorize this too, is that the DNA sequence here, this thing where the LAC-I normally binds, this has been mutated. Okay, so now we have LAC-I mutated. It can't bind the operator, and the operator itself is mutated in the site where the LAC-I protein normally binds. So we have got two reasons that this is going to be constitutively expressed. And that's what that C means. Constitutive. That's how we spell that, right? Yep. Okay, constitutively expressed. That means LAC-I cannot bind to the operator, and as a result, RNA polymerase is just going to keep transcribing those structural genes. So let's put another X here. Okay, so we have 4 more to go. Now this one is a little tricky because we're dealing with these F' plasmids, and we haven't talked that much about plasmids yet in this course. So what are plasmids? So essentially, it's easiest, I think, to think of them as extra chromosomes. So E. coli has a chromosome, 1 chromosome, and it's 4.6 million bases long. Well, they can have extra DNA in here as plasmids, and we're going to think of it as an extra chromosome. They can be very small. They can be a couple thousand base pairs long. And we're calling this particular one, F', and there's a reason for that, and we're not going to worry about the details of why it's called F'. But there can be genes on F' plasmid here. I'm going to call this a plasmid again. Let me change back to yellow. I think it's a little easier to see. Okay, plasmid. And there can be extra genes on the plasmid. In this case, we have an extra copy. Now we're going to look at this genotype right here. Here, let me put a new copy of the table here. Now we're going to look at this one right here. Okay, so copy of the operator right here on the plasmid. So this is different than the operon that is present over here. So over here we have what's shown there on the left of that genotype. We have I minus, OC, and Z plus. Okay, so what does this mean? Exactly what's going on here. So the operon is still at its normal place over here on in the chromosome, in the normal chromosome, where we have LAC-I, right? LAC-I followed by a little gap there, then the start of the operon, where we have the promoter, LAC-O, LAC-Z, LAC-Y, LAC-A. So all this stuff is still over here on this chromosome over here. But over here on the plasmid, well, we just have an extra copy of LAC-O. And this one, let's look at the genotype. This one over here is LAC-O plus, and this LAC-O over here, the normal spot, normal location on the chromosome, is LAC-OC. And also, LAC-I over here is actually LAC-I negative. Okay, so we have a mutant version of LAC-I, we have the constitutive version of LAC-O over here, and we have a normal copy of LAC-O on the plasmid. So our goal is to figure out if these genes right here at the normal location are going to be constitutively expressed, are they going to be repressed, never expressed, or are they inducible, like the normal situation? You add lactose and then they're expressed. So it turns out that, okay, so let's, the inhibitor, right, LAC-I is mutated, so it cannot bind either this LAC-O here or this LAC-O up here. It can bind neither one because it's mutated. This LAC-O up here has the binding site, so if it wasn't mutated, it could bind up there. But this one right here, it doesn't even have it. It's the mutant version. So RNA polymerase is going to come in here, bind at the promoter, and express all of these structural genes. Okay, so they're going to be constitutively expressed. There's nothing to stop RNA polymerase from binding at the normal location and keep expressing these structural genes Z, Y, and A. This one right here, this copy of LAC-O on the plasmid has no effect on the constitutive phenotype. It's just hanging out there on the plasmid, doing nothing. It's doing no good at all. It's useless. So we're going to put constitutive for this one right here. Okay, let's take a look at the next one. So this one, I'm going to move down here a bit. We have, what do we have? Okay, at the normal location, I'll just sketch this really quick. This is the E. coli chromosome over here, and then this is the F factor. And over here on the chromosome, we have I plus, OC, and Z plus, and over here we have just a normal copy of the operator. Okay, so now, okay, what do I sketch? Really quick, sketch the operon again. It's going to be kind of messy. P, O, Z, Y, A. Okay, and then I'll just put O over here for the F prime plasmid. So this is the wild type, so we're going to put that DNA binding site, the LAC-I binding site on there. This one is mutated, so I'm not going to put the no binding site. Or maybe I'll just change it. I'll put a right side up triangle here. Okay, no binding site here. It's broken, mutated. So that's C. This is I plus. Okay, so this means that in this E. coli cell, we have a normal LAC-I repressor. But the LAC-I repressor cannot bind to the operator, right? Because this DNA binding site was mutated. The way I've drawn it here, it doesn't fit, so that works. This can bind over here to the operator on the plasmid. So, okay, well, if this was part of an operon, it could block transcription of the genes after the operator, but it's not part of an operon, it's all by itself. So if lactose was around, no, don't even worry about lactose being around. This LAC-I right here is going to be bound to this operator when lactose isn't around, and it's going to have no effect on expression of anything. It can never bind here, right? Because this is the LAC-O constitutive version of this gene here. So RNA polymerase is always going to express the structural genes. So we're going to go with constitutive for this one too. We haven't seen repressed yet, have we? So I got a feeling we're going to see repressed next. Okay, so let's take a look at the next one. Let's see, I think, okay, over on the E. coli chromosome and on the plasmid, okay, so E. coli chromosome, we have IS, O plus, and Z plus. So normal copies of the operator in the structural gene Z, and we have a different mutation of the LAC-I gene. I'll explain what that is in a second. In the plasmid, oh, we have nothing. We don't even have a plasmid this time, so hopefully this will be a little easier. So quickly sketch the operon again. Getting better at this. Okay, this is going to be IS, mutated version of I. This is going to be the promoter, operator, normal operator, so I'm going to put, I'll say these are all wild type here. And I'm putting that DNA binding site, normal binding site there. there's no plasmid, so we only need to worry about the operon. Okay, so this is what lac I normally looks like. This is how we've been diagramming it. So this is lac I plus, with the DNA binding site, the operator binding site, and the lactose binding site. OP and lac for lactose. So we have lac I minus, which is mutated up there at the operator binding site, so it can never bind. It doesn't matter whether lactose is here or not, but the lactose binding site is okay. Okay, now we have lac IS. And lac IS is for a super repressor. S is for super repressor. Now that means the lactose binding site is broken, but the operator binding site is intact. So it's always going to be bound. This is always going to be bound to the operator. Even when lactose is around, lac IS is always bound there. Okay, that means even if lactose is present, the cell, E. coli, would want to express these genes. RNA polymerase is going to try and transcribe, but for the most part it's blocked. It's not going to be able to transcribe those. Whether lactose is there or not, no transcription. So this is going to be the repressed state. The lac operon is always repressed. So what about, so this next genotype right here is the same thing, except now we're going to add something to the E. coli cell. We are going to add the plasmid, and we are going to put a normal copy of the lac I repressor. So give me my pen. There, okay, there I go. I plus. Okay. So what's going to happen now? Now the cell encodes both this one right here from the normal E. coli chromosome, and it encodes this one right here on the plasmid. So the cell is going to express both of these at the same time, and both of these proteins are going to be floating around in the cell. And we can add lactose, okay, and then lactose could bind here, and then this couldn't bind the repressor anymore. But this one over here, this one's always made, whether or not lactose is there, this is going to be bound to the operon, to the operator of the lac operon. So it's always going to be blocking transcription of the operon, no matter whether lactose is there or not. And it doesn't even matter what this one is doing. This one is responding to lactose and undergoing allosteric shifts when lactose is there and when it's not there. But because this lac IS is also in the is on the E. coli chromosome, and it's always expressed, it's always going to be bound, and okay, this operon's never going to be transcribed. So this is also going to be a repressed state. Okay. So again, come see me if you still have trouble understanding how this question, or the solutions to this question. Okay, I'm going to move on to 16.6 now.